Good morning, everyone, and welcome to this week's Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar. Before passing the baton for the introduction of today's speaker, I want to remind everybody about next week's upcoming program. So next week on November 19th, same time, we'll have Dr. Doug Johnson, who's an associate professor of medicine and the clinical director of the melanoma program at the Ingram Cancer Center at Vanderbilt University. And Dr. Johnson will be talking about virtual pioneers and dissecting toxicities of immune checkpoint inhibitors. So without further ado, it's my great pleasure to introduce Dr. Brittany Howe, who will introduce today's speaker, Dr. Ralph Adolphs. Brittany, it's all yours.
Wonderful. Thank you, Mike. So it is really a distinct pleasure to introduce Dr. Ralph Adolphs as our Pioneers in Biomedical Research speaker today. So I'm not going to focus too much on his background, academically speaking, because I feel like if people are interested, you know how to Google just as well as I do. And I'm sure if you want to reach out to Dr. Adolphs, he can answer your questions there. But just to give you a brief background of his work, so he got his undergraduate degrees and a master's degree from Stanford, and then he got his Ph.D. in neurobiology from Caltech. But things, I think, really got exciting when he moved to the University of Iowa to do a postdoc with Antonio Damasio, where he described some of the impacts on emotion processing in a patient with bilateral amygdala lesions. And so this really started this amazing work that he's going to talk about today, but really gave us some insight into what this specific structure is doing in the brain, or at least one of the many things that it's doing, right? So from there, he was an assistant professor at Iowa State, and then he moved to Caltech, where he is currently. He's the director of Caltech's Brain Imaging Center, the chair of the Caltech Institutional Review Board, which I think is pretty notable, as well as director of Caltech's Conti Center for the Neurobiology of Social Decision-Making. He's published many, many articles, nearly 300. And what I think is really remarkable is that he's published in things as, small as single cell recordings, all the way up through behaviors, not just in humans, but in lots of animal models, including some unique ones like barn owls, which I think is super cool. But I, again, wanted to not focus on those things because you can find those out, but I actually did a little bit of digging. So it just so happens that my postdoc mentor, Jed Elison, was one of Ralph's postdocs. And so I reached out to him to ask if there was anything that he would like to share about Ralph, or he would be willing to share. And he told me 4 things. So one, that Ralph likes spicy Northern Thai food. He likes a good California IPA, and that he likes running up and down mountains. But I think the most important thing to emphasize here is that Jed also said, and these are Jed's words, that Ralph was perhaps the most conscientious, generous, and capable mentors that he's come in contact with, which I think is, of course, I know Jed well, and that means an awful lot. But anyway, moving on to his talk. So again, Ralph's career was really built on addressing complex questions using lots of cool models and levels of analyses. And I'm hoping that we will hear a little bit about that. I'm sure we will. But overall, he's trying to understand how our psychological understanding of human social behavior really arises from neural processes at many different levels. So whether you're talking about the systems level or the circuit level. Today, he's going to focus on the translational branch of his research, looking at children with autism or people with autism spectrum disorders. And so with that, I'll stop talking and let Dr. Adolphs take us into his work. Thank you.
Thanks for the kind words, Brittany, and channeling Jed's kind words. Yeah, Jed was great to have in the lab. We still loosely collaborate with him. Just to double check, so the format is I have until about 9 or so, or sorry, I have about an hour. I'm 3 hours behind you guys, so.
Sure. So if we could leave, roughly 5 to 10 minutes for questions, and I'll moderate the discussion at the end.
Okay, great. And I have my chat open here. I can't see any of the other people on here, but if I don't know if they can put things in there. But if there's anything that's really unclear, or some technical issues throughout, people, including you, should feel free to either just interrupt by saying something if you're able, or put something in the chat, and I can kind of monitor it. So thanks again for inviting me. I had initially intended to visit you guys in person. It turned out to be logistically too hard to do, so here we are on Zoom. And just to say a little bit more about something that you mentioned in your introduction, Brittany, and something that Mike Friedlander and I spoke about right before this this talk. So I did my PhD in an animal model, the auditory brainstem of barn owls, and then switched when I went to the University of Iowa to work with Antonio Damasio as a postdoc, and then subsequently stayed on as junior faculty to do human cognitive neuroscience. But that, that theme, I think many of us share, the theme of trying to find some convergence or links, some crosstalk between work in non-human animals and work in human animals. And indeed, the building that I'm currently in here, which is part of the Chen Neuroscience Institute at Caltech, which is relatively new, tries to do exactly that. So we just moved my lab here recently in the middle of COVID, starting to slowly get human subjects research back up and going. But I'm the only lab in this building, and there's a lot of them, that does human work. There's work in flies, in rodents, et cetera. I have people like David Anderson, with whom I wrote a book on emotion recently, right next door. And one thing that we're trying to do is indeed to find some common ground and some scientific questions, ideally that we could collaborate on, that we could address, in rodents and in humans. It's hard to do, but that is something that we're trying to do in this building at Caltech. I'm not going to talk about animals, non-human animals, in my talk. Let me share my screen with you and make sure that you can all see it. So here's the talk. Again, let me know, interrupt me if you cannot hear me well or you cannot see this. So I'm going to give you an overview of work in the lab that looks at the intersection of these three topics: autism, face perception, and the amygdala. And roughly, it's going to be divided into three parts. I'm going to begin sort of giving an overview of some of the work that we're doing. These are in high-functioning adults with autism. So right at the outset, this is not a representative sample of people with autism spectrum disorder, but it's one that we've decided to focus on for a number of reasons. One is just practical, that a lot of the tasks required require relatively high-functioning adolescents or adults, as opposed to doing work in children or infants, which is, of course, important and more representative since autism is a developmental disorder. Then talk about the amygdala and tie that back to autism, and then finish up by saying just a little bit about some methods that we've been focusing on that try to take some of this work to the next level, and in particular, try to have a stronger causal model for what it is that the amygdala might do than the other methods allow. And so for each of these three components of the talk, there's some challenges that I'll tell you a little bit about that we're trying to address in our work. With respect to autism, we've put in a lot of work in assessing in great detail, doing ADOS here in the lab, getting people to be reliable and consistent on that, which takes a huge amount of work, and then carefully matching people with and without autism on as many psychological variables as possible, and trying to address, this huge topic that a lot of folks are interested in, the heterogeneity that you see in autism. In the ideal case, to parse out possible subtypes of autism, for instance. And these are, there's other challenges that we're not meeting. Just to jump ahead, one big one throughout our work and throughout all of my research with humans, pretty much, has been small sample size. And so we are limited by a relatively small number of subjects whom we try to investigate in greater depth. That sort of characterizes a lot of the research in the lab. For the amygdala, we've been trying to get better spatial resolution and use methods other than bold fMRI. And then for the methods here, I'm going to talk a little bit about causality. So right at the outset, just to acknowledge some of the many people with whom we collaborate, the ones most relevant to what I'll be telling you about. These are folks here at Caltech in the lab. The ones in parentheses have moved on to various places. But Lynn Paul in particular, who's actually staff and a clinical psychologist by background, has been in my lab forever, right since I came here about 2004, 2005. Does a lot of work in autism, and she herself also has grants and leads research on a very interesting related developmental disorder, which is agenesis of the corpus callosum. Many of these other folks, Dorit Kleeman is now at the University of Iowa, just moved there as junior faculty, used to be a postdoc in the lab, and Shua Wang who's at WashU now as junior faculty, also used to be a postdoc in the lab, did a lot of work on autism as well. We collaborate with a number of other institutions, and for the work in autism in particular, we collaborate with another one of my former postdocs. You build up this, this network of people all over the place that eventually have their own labs that used to be in your lab as graduate students or postdocs. But Dan Kennedy at Indiana U has been collaborating with us, and what we've been trying to do in a lot of our autism work is to actually have two separate samples, and discovery sample or replication sample, and to use parallel stimuli and do parallel kinds of experiments at Indiana and at Caltech on our respective autism samples. Cedars-Sinai Medical Center, University of Iowa, these are hospitals where we collaborate with the folks here. Here, Yuki Oya used to be with me, and Uli Wuteshauser used to be a postdoc with me, and then Adam and Matt are the neurosurgeons. These are resources for single unit recordings in epilepsy patients where we can record from structures like the amygdala that I'll tell you about a little later. We study all of these populations here and these are all clinical populations where one of the things we try to do is to find some convergence between them. So for example, we make some comparison between people with autism, which of course is not neurologically, but psychiatrically defined, with neurological populations like amygdala lesions, folks with agenesis of the corpus callosum, and then there's several other very interesting neurological populations that we study. One thing that cuts across all of this is the issue of plasticity and compensation. And so we've been very interested in questions like what happens if you have a hemispherectomy early in life, or what happens if you're born without a corpus callosum in terms of, for example, the integrity of functional brain networks. And in both cases, we published on this, the answer is they look remarkably normal. So there's a remarkable amount of compensation that happens in some of these developmental disorders that we hope may also give us some insight into what happens in autism, which is of course a pervasive developmental disorder as well, that was first sort of studied in detail more or less contemporaneously by Kanner and Asperger, although others had used the term before then. And although it's gone through various cycles and originally used to be a distinction between Asperger's and autism, now in DSM-5, as you probably know, it's autism spectrum disorder, with the realization that this is really a collection of disorders. It's a broad spectrum that you can characterize to some extent parametrically with respect to intellectual functioning, delayed language onset, etc., rather than putting it necessarily into categories. But one of the core features, or three of the core features that are listed here, are this desire for sameness, rigid behaviors, repetitive behaviors, often delayed language onset, and then the aspect of the phenotype that my lab has focused on most, which is difficulties in social cognition and social behavior, difficulties in attending to social events, difficulties in making inferences about what goes on in the minds of people around you. And that's the collection of psychological processes that we focused on. It's a hugely complex disease. It's highly heritable. So there's a strong genetic predisposition that interacts with environmental influences early on in development already, and this unfolds throughout early development, eventually giving rise to some neural signature. One that's been studied a lot is some atypical connectivity between brain regions, but there's also abnormal brain function in many brain regions. So this is the level that we are trying to understand how this then causes the altered social behavior and social cognition that is part of the phenotype that's the basis for the diagnosis in the first place. We're not studying any of these other components, even though these are, of course, extremely important, and probably most people working in autism are looking at many of these components and looking at the genetics, but we don't do that in our lab. So what's been found at that level? Well, there's a large number of studies, but going back to sort of some of the original ones, basically, if you look across functional neuroimaging studies and you ask, what do people find that looks atypical in the brains of people with autism, it can be summarized by these two lines here. Here's some of the studies in a little more detail, but it's increased variability, both across subjects, across people with autism, so greater individual differences between subject variability, as well as within subjects. And that's sort of where the story has been for quite a while. And the challenge then has been backing out anything specific there. There's no shortage of hypotheses and there's no shortage of brain structures that people have looked at or brain networks, but finding something reliable or something specific has been difficult. And one story that I will mention in just a second, where we think we can get a purchase, is that although indeed there is heterogeneity between subjects with autism, and again, the hope is that if you have a large enough sample and if you have the right dependent measures, you might be able to back out subtypes of autism from this between subject variability. We think that in fact, with the right measures, you can get a handle on within subject variability, and this is not often assessed, so that this is actually could be relatively stable within a subject, depending on what the dependent measures is. And then this is one of our early studies where we indeed found that the differences between people with autism and matched controls was largely driven by outliers or subpopulations, that there were many individuals with autism in this functional neuroimaging study that looked identical to controls, but then there was a smaller group that was different. In terms of, one of the main workhorse dependent measures that people have used and that we also looked at in the past, it's the correlations between brain regions that are thought to be a signature of how information is passed between distal brain regions, the so-called functional connectivity matrix. And so what this shows here is if you parcellate a brain and you just put somebody in a scanner and you get resting state signals, you have a time varying signal in this brain region, and this brain region, and this brain region. And if you look at the simple pairwise Pearson correlation between all pairs of brain regions, you get a correlation matrix, a triangle symmetric matrix, like what's shown here, with the color encoding the strength of the correlation. And for instance, here you find these are homotopic connections between corresponding parcels in the left and the right cerebral hemisphere, so they're strongly coupled. And then there's variable correlations between other brain regions, and from this you can use clustering methods to derive resting state brain networks, functional networks that are now studied in great detail. If you look at the literature on this, there are a lot of studies and lots of hypotheses claiming that there is abnormal, atypical resting state functional connectivity in autism, perhaps reduced long-range connectivity, but there's very little reliability or in those studies. When we looked at this ourselves, here's the functional connectivity matrix in autism, in our group of people with autism, in our controls. And this is just a plot across all of these correlations between plotting controls versus autism. The bottom line of this study that we published some years ago now was that when we matched people with autism and controls, so they were identical age, of course, same sex, similar IQ, we really couldn't find any difference here. This on the right here, this just plots across all the different brain parcels any difference in connectivity between autism versus controls. And there's a few that pass this threshold here, but no more than you would expect by chance. So we were not able to find any difference. This is just a histogram of the correlations across this entire matrix between autism and controls. There are differences if you don't control for confounds. And so, for instance, if you don't control for motion in great detail, you do find that there are differences if you partition the group not by diagnosis, autism versus controls, but how much they move in the scanner instead. Then indeed you do find differences. So what we found, and I think what several other groups have found, that the more carefully you match with respect to intellectual functioning, with respect to movement and so forth, your groups, the less of a difference there is, and you're not left with much eventually. A lot of work has been put into resting state fMRI, and we and many others, have put out databases like the Abide database, for instance, which is resting state fMRI data from many different sites. Caltech is one of them. There are many others. And when people have looked at this in detail, it's varied. There's no shortage of studies. But there's studies, for instance, from Lisa Birch and Dan Kennedy, our collaborators at Indiana University, that really you cannot reliably find a difference using resting state fMRI between autism and controls. In the Abide data set, in that paper that I just mentioned, you can tell what the performance site is, which university the data came from, but you cannot distinguish autism versus controls. And resting state, if you think about it, is probably not an ideal state. And so in this recent TICS opinion from Emily Finn, she makes the point that indeed we should probably use other kinds of stimuli. And the stimuli that we and many other people have used are stimuli that have a lot of advantages. They're videos, movies, video clips, movie trailers that have one advantage is that they keep people very attentive. It's easy to keep people awake and focused on it. They're very rich in the numbers, in the different types of features that you can now annotate largely with automated methods. So there's lots of faces, there's hands movements, there's this tone of voice. So you're getting a very rich feature set. And because it's the same stimulus that's driving all of these different brains, you get very, very reliable brain responses from your subjects. So in one study that we've been working on and analyzing for quite a while, this is the collaboration between Caltech and Indiana University that I mentioned. And we have these sample sizes of people with autism. Again, that's one major limitation of the work we've been doing. These are small sample sizes. These are all, again, high-functioning adults. At Caltech and at Indiana University, here's the sample sizes for subjects with autism and controls. We show them lots of these different videos, and we get both brain imaging and eye tracking data. So you have, basically, this is the experiment. I don't know exactly why, but at the time when he was a postdoc in my office, Dan Kennedy latched onto the sitcom The Office, so that all of the main stimuli are episodes of The Office. You would line a scanner, you would watch this with sound, and then you would have a time varying change in your brain activation that's driven by the stimulus. Everybody sees the same movie, and so as you might expect, what you find is actually pretty reliable activation. So what this plots here, each blue line is a control subject, and the black is their mean. And on the y-axis here, we're plotting just the normalized activation that you would measure as a function of time on the x-axis in visual cortex. So if you just extract this from visual cortex, what you find is that this is fairly reliable. Each subject differs somewhat from the rest. The data are noisy, these are fMRI data, but nonetheless, every once in a while, there's something very bright, something darker in the movie. And since everybody watches the same Office sitcom, you get very reliable brain activations. And same thing in autism. And in fact, if you, for visual cortex, just superimpose autism and control time courses when subjects are watching the same video stimulus, you get a strong, very similar time courses. So many people have looked at these inter-subject correlations. Uri Hasson at Princeton has probably done some of the leading work on this. And you can ask to what extent are regions of the brain correlated in how they're driven by this stimulus. So here, visual cortex, or if you were early on in the LGN or the retina, the visual input is identical. They're seeing the same movie. So you're going to get the same activation time course across everybody. As we march higher up in the brain, there might then be, an episode in this video that we're watching that I think is really funny and you think is really dumb and not funny. And so there, our time courses would diverge. That wouldn't happen in the retina or the LGN or visual cortex, but higher up, perhaps somewhere in prefrontal cortex. So the thought is that we can, to some extent, parse where in processing of this rich social stimulus might be different in people with autism than in controls using these intersubject correlation type of approaches. And that's something that we've been working on and are still working on. It's a large data set, and I don't have an answer to the question or to the strategy that I just posed to you. Here are the data. This is the inter-subject correlation just for the first 1,000 TRs here of the group tested in Indiana and at Caltech. And it just makes the point that across different testing sites, different scanners, different subject samples, if you have the same video stimulus, you can evoke very reliable, similar inter-subject correlations. So one reason we've been using this, rather than, for instance, resting state, is that it allows us to get highly reliable cross-site results here, and then ask the question when we look in detail, what might be different between controls and autism? There is something different, and so what's shown in the bottom here for 4 different videos as compared to resting state is the accuracy with which you can classify autism as distinct from controls from using all of the different brain parcels and looking at inter-subject correlations. So we just train up a logistic regression, autism versus controls, using ISC and all the different brain parcels, and we hold out a subset of the subjects and ask how well can we predict that from that logistic regression model. And the accuracy is these are all these dots here. So the accuracies are fairly high, even if you subsample the data. So if you decimate the data and you only use about 5 to 10% of your data samples, you still get pretty good classification. That's what these little asterisks and plus signs are. So we can tell subject group, autism versus control, apart from even a fairly small subset of the inter-subject correlations that are evoked by watching movies. The next question then is, okay, where in the brain is it? And at what time in the movie, at what particular epochs is it? And that's what we're working on next. And I don't have any results to show there, but there are differences at particular points in time and at particular places in the brain. So we're using windowed ISCs, moving a window across the entire video and looking at particular brain regions to ask, where do these correlations evoked by the movie differ the most in time and in brain anatomical location between autism and controls? You can do more with this. That's more like Jack Gallen style encoding models, just a sort of very, very simple version of it. That is, these movies have a lot of features, some low-level ones, brightness, visual motion, and so forth, and semantic ones that often you cannot get in an automated way, but you need mTurkers to watch the movie and give you. So one is how socially awkward the video is. And The Office is, as far as I can tell, pretty much all socially awkward, but this is the time-varying social awkwardness of this of the video as rated by an independent group of online subjects. So it varies over time. There's some really awkward moments, some not. If you just use this as a regressor, you can also find differences between autism versus controls. So this gets at the kind of question I was hinting at. If we go to higher level brain regions and ask, well, what is it about the video? What higher level semantic features, like social awkwardness, as opposed to just brightness or darkness, might distinguish groups? Then we can find regions like the right TPJ, posterior STS in this particular example here, where indeed you find differences between autism and controls. They're small, and so what's plotted here is from this paper, SCAN, is the actual weight of the evidence in favor of a group difference, autism versus controls. And right TPJ or STS, there's some, but weak. None of these other ROIs show a difference. The different sizes of the circles here are different sizes of spheres that we planted into the ROIs that are indicated here. Okay, I'm very quickly going to go through this one here. This is a study that we're still working on. It's predicated on a task that tries to back out one of the main psychological processes that surely come into play when you watch a video like The Office, among many, but it's the one that certainly in the literature has been studied the most, which is something called mentalizing or theory of mind. It's basically making inferences about people's internal states, their beliefs or thoughts or intentions or emotions from watching them, either move around or looking at their face or looking at hand movements. And so the task that Bob Spunt, who was a postdoc in my lab, developed is this level of inference or why-how task that shows you a picture of somebody doing something from which you can either just describe the action. So in one version of the task, you just ask, is the person doing some particular action, like looking at the camera? And in the other case, you see the same stimulus, the same person, but you're asked to make a judgment about the hidden latent variable that's inside this person that's causing them to behave in this way. That is, are they self-doubtful in this particular example? So you use this task that has a very simple contrast that looks at a level that tries to isolate this cognitive process, so making social inferences about people's internal states. And when you do that, like in this original neuroimage papers, a lot, and Bob has published a lot on this, a lot of standard univariate analyses reveal in individuals, at the level of individual subjects, a reliable network of structures that includes, amongst other places, dorsomedial prefrontal cortex, and this region, posterior STS and TPJ, that has long been highlighted by people like Rebecca Sachs and many others as being involved in this kind of process, this theory of mind inference. So here we had 4 different samples, a discovery sample, 2 replication samples, and a sample with autism. And in all of these, in this particular example here, going along with a multivariate searchlight all over the brain, we find this network of brain structures, the particular ROIs listed here, where you find this contrast. So they seem to be, they're activated by making mental inferences about people. And one thing that we found in this particular study was that people with autism were different, and there was a correlation between ADOS scores as well as autism quotient and the signal in these particular ROIs. We also had a separate task here, or separate questionnaire, where we looked at the number of social contacts from the social network index, and that was also correlated with signal in this region. So I bring this study up and just go over it very quickly as one example where we're trying to correlate signal that's related to a particular psychological function, in this case, making mental inferences about people, and processes that are relevant, or measures that are relevant to autism, number of social contacts, AQ, ADOS scores. The main problem with this approach, even though we have these different replication samples, is that the sample sizes are very small. So again, coming back to one main limitation of a lot of our studies, we don't have that many people, certainly with autism, and even in our matched control groups, the sample sizes are small. One way we're trying to solve that is to have discovery sample, replication samples, and see if this replicates across different samples, or as in the case of collaboration with Dan Kennedy at Indiana University, indeed replicates across sites. So that's one main methodological theme, that we're working with high functioning individuals with autism. We're doing labor intensive tasks. They watch lots of videos. The sample sizes are small. We're not looking at hundreds of subjects. And so if you want to correlate some signal from fMRI with individual differences, most people would tell you, well, you need hundreds of subjects to back out anything reliable. We don't have that. And so instead, what we're trying to see is if we can replicate effects in independent samples. So I mentioned one of the main processes that's been studied a lot in the literature that is thought probably both in a developmental context and online to contribute to the atypical social cognition and social behavior that's part of the phenotype for autism spectrum disorder. Another one that's been looked at a lot is attention, and in particular in a developmental context, but also online. You could imagine that if you do not attend normally to social cues, if you do not look normally at people's faces, that this would also give rise to atypical cognition about those social stimuli, because you're not picking up the relevant social information in the first place. And so in the scanner, we, like most others, use eye tracking in the scanner. And I'm always impressed by this is a speeded up version of a couple of autism subjects in red and controls in blue, just showing you another dependent measure that's actually extremely rich that you get in addition to the fMRI, which is eye tracking. And it's more than just gaze. So you certainly get to see where people are fixating on the movie, but you also get their pupil dilation, you get their blinks. And so we've been asking, what can we do with eye tracking to look at autism. It's another big field. So in addition to fMRI, there are an even larger number of studies using eye tracking. Just to give you an example, just a feel for the kind of data that you get. This is not in autism. This is building on a data set from people in Sweden. So Dan Kennedy collaborated with these folks here. This was a published paper on still images. But this is a couple of hundred children who are either identical twins in red, fraternal twins in blue, or unrelated in gray, who are watching, not The Office, but a Swedish popular sitcom. And so they're watching this movie. The dependent measure is eye tracking. Here in the lower left, you can see each of these 100 children, participants in the study, where they're fixating at various points in time. And this green line moving along here is the time. And these curves here plot the correlation between where people are fixating. And so if there's only, I don't know, somewhere over here, a single face or two faces, everybody looks at the face. And so the correlation is high because there's nothing competing for visual attention. Whereas down here, there's a number of faces, the correlation is lower. But the point here is that you can use eye tracking also, similar kind of idea to the intersubject correlations from the fMRI. Because you have a common stimulus, you can look at the correlations between where people are looking, and you can ask, are there some subjects that fall outside of where everybody else is looking? In this particular study here, this shows that genetically related individuals look similarly at social information. So even though the curves are all fairly close together, you can see that the red curve, which is the identical twins, has stronger correlations than the other curves. And this has also been studied in relation to autism, that there's a strong genetic component to where people fixate. And autism is highly heritable, and so you would think that there has to be some story to tell about how individuals with autism might fixate these stimuli differently, in part a difference that could be genetically traced. But so coming back to the stimulus that we used in the scanner, we have the Office video, we're getting these brain imaging data, and we can ask, where do people fixate on each frame of the video? And when you do that and use a very simple measure here, just the contrast in each frame of the video, you find that indeed there is a group difference, but it looks like what I mentioned at the outset of my talk, which is that many individuals with autism fixate regions of the video, of this Office video, that have high or low contrast, that looks the same as the controlled subjects. So they're not different from controlled subjects. But then there's this tail here of individuals with autism that look different from the controls. And this is what we found in essentially every study. There's a large proportion of people with autism that we cannot distinguish from controls. And if we train a classifier in eye tracking data, if we train a classifier in the fMRI data, that classifier confuses these autism subjects with controls. And then there's a portion of people with autism that the classifier tells apart as people with autism, and they're all kind of different. And so that's sort of where the story currently is. And again, the hope is that this group here of people with autism that are indeed different from controls, if we had a large enough sample, might perhaps fall out as distinct subtypes. For the sake of time, I'm going to skip this just to say that we can build much more sophisticated models to analyze these eye tracking data, just like we're doing for the fMRI data. So it's not just like brightness or contrast in the image. And it's not just social awkwardness that might be rated, but everything. So in the ideal model, we put everything together and we have both low-level features, brightness, contrast, visual motion, etc., object level features, is there a face, is there text, etc., and semantic level features. We put them all together in a model, and we use models like this both for the analysis of the fMRI data and for the analysis of the eye tracking data. In this Neuron paper that Chuo had some years ago, we used it to analyze eye tracking data, and we found that indeed people with autism, the red curves as a function of fixation order plotted here, differ where they fixate from controls on a large number of different features. They tend to fixate more at the background, and their fixation, where they fixate, is driven more by pixel level features, just local contrast, visual emotion, and they are less affected by semantic level features and object level features. So from this study and from everything else we've done so far, again, we can classify, it's very similar to the fMRI story. We can classify people with autism from their eye tracking data as distinct from controls, but we can't pinpoint it to a specific feature in the video. It's not that they're not just looking at faces, they're not looking at any object level or semantic level feature typically, and it varies. So the atypical gaze, the effect, the weight of the features, how they contribute to atypical gaze, is distributed across lots of different features. Despite all of that, and this is a recent study from the same data set, or analysis from the same data set, what we find is that individuals are quite reliable. So what's plotted here is each dot here is one subject. There's a lot of information represented here, not all of which is essential to the story, but these are autism on the left and control subjects tested at Indiana University and Caltech. The details don't really matter. The point is each dot here is a subject, and what we're plotting is where they're looking on one video tested on one day. So this is episode A of The Office versus another episode of The Office on a different day. So two different stimuli on two different days. And for the same subject, we ask how much time are they spending looking at faces or looking at eyes, or just a heat map correlation analysis that's not based on any particular features. And what we find is that across the two different stimuli and the two different testing dates, there's a strong correlation. Those individuals that don't look at faces in video A also don't look at faces in video B. And this is true across both autism and controls. And in fact, this reliability in how they fixate different features in the stimuli is the same across controls and autism. So the bottom line from this is, even though, as I've been telling you, people with autism show considerable heterogeneity, and there's this long tail of individuals with autism that differ in varying ways from the controls, within a subject across different time points, we are getting reliable signatures. And this is true in the brain imaging data as well. So the hope is that we can indeed back out something here, because we have a handle on the within subject variability across time, which is stable. It's a trait-like temporally stable signature that we're getting in terms of how they differ from other subjects in the features that they fixate in this particular paper. I'm going to skip this one since I see I will shortly run out of time, and move on quickly to the amygdala. So the amygdala is a structure that's very densely connected with many other brain regions. This is just a multidimensional scaling of connectivity between the amygdala and many other brain regions in the cat from this paper here. It's been studied a lot in relation to autism. It gets highly processed visual input from temporal cortex into the lateral nucleus, and then there's a lot of internal processing that allows the amygdala to bias how you make decisions, how you encode information into memory, and what kinds of emotional responses are triggered by stimuli. It's very complex internally, and so one caveat to all the studies with fMRI is that we're unable to resolve the details of these different cell populations that are shown in this particular picture. I'm going to skip all of this. As Brittany alluded to, I did a lot of work on the amygdala, especially when I was at the University of Iowa looking at patients with amygdala lesions. One upshot of that further motivates making comparisons between amygdala lesions and autism, or looking at the amygdala in autism, in addition to lots of work by other people, is eye tracking. And so if you ask how do people fixate faces, people with autism on average look less at the eyes than do controls. This is the difference. A rare subject who we studied at great depth, who has selective amygdala lesions, also doesn't look at the eyes. And in this whole list of papers, we dug in detail into how people process faces, and there's some overlap in autism and amygdala lesions. It's not the same, but there's some similarity. They don't seem to process information about the eyes normally. They don't fixate the eyes normally. To look at this in some detail, I want to show you one last main study here, which doesn't use fMRI, in part for the reasons that I mentioned. Signal in the amygdala is terrible. Most studies of amygdala fMRI that are in the literature will, I can guarantee you, not replicate. If you look at big databases, even Human Connectome Project, if you ask what's the temporal SNR in the amygdala, it's terrible. What's the signal in the Abide data set in the amygdala? It's terrible. There's too much dropout. There's lots of motion problems and so forth. Anyway, so, which is not to say you cannot image the amygdala, but it's far from ideal. Instead, we took advantage of our collaborations with Cedars-Sinai Medical Center, University of Iowa, where we can collaborate with neurosurgeons who have implanted electrodes directly into the amygdala. These are patients with epilepsy. For epilepsy monitoring, they have these electrodes in there, and we can record single units from the amygdala. So the setup is there's an awake, behaving, high-functioning patient here, with a big bandage on his head, and he has depth electrodes in the amygdala, typically bilaterally. And we can show him videos and pictures and faces with concurrent eye tracking while we record ECOG and single units. Skip this. The study that I want to present here briefly that we published in Neuron a couple of years ago took advantage of the comorbidity between epilepsy and autism. And so through these epilepsy centers, they would get maybe a patient or two a month with depth electrodes. We would do our studies on face processing. And every once in a while, by chance, you might think, well, maybe we will get a patient who has epilepsy and depth electrodes who also has a diagnosis of autism. And we got two of those, these ones up here, with the ADOS on them. And so they met criteria for autism. And we compared those to the controls here. These are not healthy individuals. These are people with epilepsy, but without autism. These are the recording sites. The blue ones are the people with epilepsy, but not autism. And the two red ones are these two case studies of people with epilepsy, depth electrodes in the amygdala, and autism. When we record neurons there, we found that the waveforms, the action potentials, and other metrics of how neurons in the amygdala respond looked normal in the people with autism and the people without autism. But what we found was that responses of neurons in the amygdala, in particular to pieces of faces, looked quite different. So the stimuli in this experiment showed people little bits, randomly sampled bits of faces on each trial, an eye, a mouth, a little bit of a nose, little pieces. And what we can then do is correlate how strong neurons in the amygdala would respond to pieces of a face to construct a so-called neuronal classification image. So in the epilepsy patients without autism, what we found was that neurons in the amygdala respond a lot to the eye region of the face and to some extent parts of the mouth. So you can imagine a face and behind it, here's the eyes, the nose, the mouth. But the eyes are particularly potent in driving amygdala neuron responses, which we found in a large number of other studies as well. By contrast, these two epilepsy patients who had autism showed that there was, these are Z-scored, showed that there was a much less of a response to the eye region of faces and a strong response to the mouth region. And these are the papers where we published this. So here we were very excited about this because this was a particularly clear finding that correlated with the eye tracking data that we find in larger groups of individuals. People with autism also do not fixate the eyes, they fixate the mouth more on average. And these two individuals, when you show little pieces of faces, this is not due to fixating a face differentially. They're shown little pieces of faces one at a time, and they can fixate all of that, move around with their eyes. We find that these neurons in the amygdala don't seem to care about the eye region as much. So at least one causal mechanism that this would support is that there's some kind of a saliency map in the amygdala. This is atypical in autism, in particular with respect to faces, that the eyes in faces are not as salient as they are in control subjects. And one consequence of that would be that individuals with autism do not attend to the eyes in faces and don't fixate there. So this ties together a little bit work in autism and one very brief snippet of work on the amygdala, in particular, the single unit recording study in people with autism, which I think is is unique. I wanted to finish up with a last, just to give you a glimpse of one particular method that we've been developing, where we try to get an even stronger causal story by doing actual intervention on the brain. And of course, there's a long history on this. If you stimulate the brain, as in this review here, an early study by Brodmann and modern day studies by essentially all of the neurosurgeons that do epilepsy surgery, they often electrically stimulate in order to ensure that the tissue that they're removing is not essential for language function, for instance. And you can evoke specific behaviors or mental phenomena from stimulating particular parts of the brain, as schematized here. And of course, this is used nowadays also with DBS, and you can stimulate particular parts of the brain in order to try to treat people with medically refractory psychiatric disorders like the ones shown here. Indeed, it has been used in a very small set of subjects also to treat people with severe autism as well. One difficulty with all of these studies is that it's difficult to know where to put these depth electrodes to stimulate in each individual, because to some extent the effects are idiosyncratic for each individual. And so one thing that we've been trying to do, both to address that translational question and also just to have an experimental tool to ask, what is the effect of actually intervening on a structure like the amygdala on the rest of the brain, is to combine direct stimulation of, in our case, mostly the amygdala, as well as any other brain structure where you have a depth electrode, with the whole brain field of view of fMRI. So we're not doing all of this stuff here. What we're doing is stimulating one brain region that activates all of those regions on which it has a causal effect. So a large chunk of brain networks that we can visualize with fMRI. Just very briefly, the paradigm is that we have a subject in a scanner. These are epilepsy patients, they have electrodes in their brain. We can acquire a whole brain volume, and then between acquisition of the brain volumes, we interleave bipolar stimulation through the depth electrodes in here. So there's a very brief burst of stimulation, and a brain volume, stimulation, brain volume. We do that for 30 seconds with stimulation on, 30 seconds stimulation off, and about 10 minutes of this gives you a contrast of what you would see with stimulation on versus off. And what you see is something that looks like this. Here's a depth electrode in this particular example in mid-cingulate cortex. And if we extract the activation from one of these distally activated regions here, you see a signal change like what's shown here, 30 seconds stimulation on, 30 seconds off. And we can then do that by bipolar stimulation in the amygdala across these subjects and evoke maps like this that give us a measure of effective connectivity between the amygdala and the rest of the brain. So these are the studies here. We just last year released initial data from 26 subjects where we have this. We're stimulating and acquiring fMRI, and this is on Open Neuro, so you can download these data and play around with them. But we think this, in the ideal case, would give us a lot of information about exactly what the effect of the amygdala on all of these different cortical regions is. And what we're doing now is trying to correlate this with individual differences on measures like the SRS or the AQ, or if we find some more rare subjects, indeed epilepsy patients who might have a diagnosis of autism. So it's what you would like to be able to do. It's still very coarse. It's not up to genetic fMRI by any means, but it's giving us at least some handle on what it looks like if we can experimentally intervene on the amygdala and visualize how that affects brain function and also task performances in these subjects. Okay, I've used up all my time, and so thanks. I will be happy to take questions and chat with all of you. If you want more information about our work, PDFs of papers, here's the website to the lab. Let me see.
Wonderful. I was waiting for my cue of the clapping. Awesome. So we have lots of questions. So let's just dive right in. Of course, thank you, Dr. Adolphs. That was wonderful. So I'm just going to go down because for those who came up with their questions first, they get them answered first. So we have the first one from Dr. Crabtree from the FBRI. And he would like to know, how do you control for attentional issues and how they impact brain region activation in response to your test videos? So, for example, differences between how well test subjects are attending to or not attending to the test videos themselves.
Yeah, I guess two main ways that we do that. One is we do use eye tracking in the scanner. So we certainly have a handle on whether or not people are, well, awake and their eyes are open and they're watching the video. We can also look and see if they fixate the video differently. If they do that, then it gets problematic how to incorporate that into the analyses, but at least in principle, you can do that, or you can pick episodes, epochs where there is no difference. So we have eye tracking. That's not identical with attention. So the other thing that we do is we ask people, we have, I skipped the slides on this, but as in some of our studies, what we do is we interrupt the video sort of every 30 seconds or minute, and we ask them questions about what just happened. And so we can quantify what they were attending to and encoding into memory with some of those questions. And we can also quantify what social inferences they were making about the characters in the video. And the slide that I skipped is a paper we're just working on, which indeed in the autism group, but not in the controls, finds a correlation between what features they were fixating, whether they were actually looking at a particular character, typically in the video, and what social inferences they were able to make from that. So those individuals with autism that don't pay attention to the characters in the video normally, that they don't fixate there typically, also are the ones that show poor understanding on these interrupted questions about what's going on socially and the intentions and internal states of those characters. So I guess the full answer to your question is we try to control for eye movements by doing concurrent eye tracking in the scanner. We ask questions on at least a subset of the videos to ensure that people are paying attention and processing it. But then indeed, we do find that for some people with autism, they do not attend to the stimuli normally in the video. And that has a consequence both on brain activation and on what it is that they can infer from the video.
So there's a quick follow-up here from Anthony Lamantia, who was curious, thinking about attention measures. Do you see any differences in activation of the cortical face areas in ASD patients versus your I always default to typically developing, to your healthy control participants?
Right. Yes, we do. It has the same trickiness associated with it as the first question, which is that at least part of the difference seems to be traceable to atypical fixations on faces of characters in the video. So yes, we have a face localizer so typically the way we do this is we have an FFA localizer and we have face patches that we then query in more detail as ROIs for particular features in the video. And so we have faces and many other features annotated in the video and can then ask how is that driving activation in that subset of in those ROIs. And it does differ in some of the people with autism, but it seems to be correlated with atypical fixations onto the faces as well. So disentangling those and really getting at the question, if you completely equated fixations and those were the same, would you then still see a difference in activation? We don't have the answer to it. It may all be derivative to atypical attention and indeed atypical fixations.
Which again, you're leading into these questions really nicely, which is nice. So Reed Montague had a question about the single cell recordings in the ASD and epilepsy patients, but they seem to actively not care about eyes, right? There seems to be almost a deactivation to the eye region, at least in those two patients. And could you expand a little bit on your interpretation of that?
Yeah, well, so the sort of deactivation in that image, that's just z-scored with respect to how these neurons respond on average. But you're right, that both in that study and for that matter with the eye tracking, this is, I didn't elaborate on this, but one difference between in eye tracking on faces between the patients with amygdala lesions and people with autism is that the patient with amygdala lesions is not averse to looking at the eyes, just not the eyes are not salient and she doesn't look there. Whereas at least in some, it varies, at least in some people with autism, it's not that they don't find the eye salient, but they're actually averse, they actually avoid the eyes. The way to distinguish those two, so in both cases, you end up not looking at the eyes, but you can distinguish those, which we have with the eye tracking. If you flash a face up, you arrange it so that the eyes in the face are where you're looking. So you have a fixation cross, person looks at the fixation cross, you flash the face up, and the eyes of the face are right where the fixation cross is. So you start at time 0 when the stimulus comes up, fixating the eyes, and you just ask what's the latency with which I saccade out of the eyes. At least some individuals with autism saccade out of the eyes more rapidly than do the controls. The patient with amygdala lesions actually does not saccade out of the eyes. She just keeps staring at where the fixation cross was. So she's not averse to looking at the eyes. It's just that they don't have any particular saliency. But that's a good point. I think there's heterogeneity in there, so that at least in some individuals with autism, you have an actual aversion. They're actually actively avoiding the eyes. And in some, the eyes are just not salient. And in some, it's a mixture of these two. But disentangling those two is indeed important. Yeah.
Okay, so I think we have time for at least one other, and I have to ask one from a graduate student, right? Because especially since they are attending. And so it feeds into another question that was asked by Reed. So thinking about this idea of social awkwardness. So so Sadia, one of our graduate students, has asked, are there differences that you've been able to note between age, race, and ethnic groups as far as social awkwardness, or at least, thinking about the office specifically? I mean, that's a very specific cultural phenomenon, right? So could you comment?
That's a great question. The quick, simple answer is no. We have not looked at those factors because we haven't had the power to do so. But it's clearly important, and it comes back to something I said at the very beginning, which is that all of this work, not just in autism, but also all of the work we've been doing in healthy individuals, is limited and not representative in many instances. With respect to autism, we do actually have some females with autism, but since autism it mostly runs in males, and especially in high functioning autism, the ratio is 8:1 or 10:1. So we have way more males than females, and we often restrict our analyses just to males for that reason. Not because we don't think that sex differences are unimportant, they're very important, and we know very little about them, especially in autism, but we just don't have the statistical power to look at that. And race, same thing. It's super important, but we just don't have the power to look at it. So the quick answer to your question is these are critical questions that need to be addressed. And I think, a lot of studies have to make more of an effort to recruit people with autism, for instance, who are older, more females, more racially diverse. We haven't looked at any of that just for reasons of statistical power. And so our results are narrow and limited to the current samples that we have.
Sure. Awesome. So then one last quick follow-up about awkwardness. Do you think if you were to use something that was less awkward, because The Office, that's its shtick, right? That's why people watch it.
Right.
Do you think that may have an impact on your results, particularly for your ASD patients?
Sure. It's all relative to, what stimuli you put in the first place. And so what we would like is to just have stimuli that have all possible features, right? Whatever that means. But what we're doing is, we are using very diverse video stimuli, which are just concatenations of movie trailers. So they have kind of everything. They're all over the place. And then we also have scrambled, both temporally and other scrambled versions of those. So those are very diverse. What those lack is an overarching storyline. So they lack that semantic feature that there's characters that you care about, and et cetera. And so there we have to pick like a sitcom. And there we can't show you, 20 or 30 or 40 different sitcoms. Although if you want on Netflix, those are available in principle to watch. I checked, but we just don't have the time to do that. So we picked one, which is The Office. But you're right, it's limited to the particular, way that the camera pans in The Office, the particular characters. It's almost all indoors. And so we're excluding a bunch of features that might be interesting to look at. we're getting those with the movie trailers, but then we're missing an overarching storyline. So we are still not sampling all of the features we would like to, but how to choose the best videos is something that we have lots of discussions in the lab, and different people come up with different sitcoms we should choose, but they all have their own limitations to some extent.
And if only you had unlimited time, right, and resources, you could just watch all of the things all the time.
Well, on that note, we are starting to do this. It's like many people have been doing. It's just like really, really deep phenotyping, really extensive scanning. And so we're scanning myself and a few other people in the lab for several months, where we're going to be watching, lots and lots of sitcoms and videos, and then at least for 2 or 3, and again, these are going to be, super small sample sizes, but 2 or 3 blue ribbon, locally available, highly motivated autism subjects, they're going to do the same. So we are going to try to get extremely detailed, data on a very small number of subjects. In the ideal case, you would be in the space where you do, hundreds of hours of scanning on hundreds of subjects. It's resource limited, of course.
Of course. Well, wonderful. So we're about out of time. Thank you again, Dr. Adolphs, for your fantastic talk. And I look forward to our meeting later.
Great. Thanks, Brittany.
Thanks, Ralph. Great talk. Really appreciate it.